Hello, today I'm going to be talking about COVID-19 testing, specifically the nucleic acid test. This project was completed by Zachary, Sara, and myself. SARS-CoV-2 is a causative agent of COVID-19. To put this simply, SARS-CoV-2 is a virus that causes the disease known as COVID-19. SARS-CoV-2 is a member of the coronavirinae subfamily, other members being SARS-CoV-1 and MERS-CoV, both of which had outbreaks in 2003 and 2012 respectively. Over 40 million people have been infected with SARS-CoV-2 worldwide, and 1.1 million people have been killed. These numbers have continued to rise as of October 21st. Like most coronaviruses, SARS-CoV-2 is a positive sense single-stranded RNA virus. However, this virus has been shown to be more infectious and deadly. In order to detect this specific virus, diagnostic laboratories have developed PCR tests and antigen tests in order to detect the virus. In this specific PowerPoint, I will be further describing the PCR test. To talk a little bit more about the structure of SARS-CoV-2, so SARS-CoV has an envelope that has envelope proteins on it, and I will further describe the importance of these specific proteins. It also has nucleocapsid proteins bound to the genome, here identified as the N protein, and it has spike proteins as well. So the spike protein in itself is used to attach the cell host receptor in order for the virus to be uptaken by the cell. So a little bit more about the biochemistry of the test. So because this is a RNA virus, we need to take an extra step in PCR, which is a reverse transcription step. So RT-PCR for short. So the necessary components to conduct this type of test to detect SARS-CoV-2, we will need Taq DNA polymerase, reverse transcriptase, DNTPs, PCR buffer, magnesium chloride, and nucleus-free water. So the Taq DNA polymerase is a thermodynamically stable molecule which could withstand the temperatures of the PCR cycling. Reverse transcriptase is used to transcribe mRNA into cDNA, which the cDNA will then further be amplified by the DNA polymerase. DNTPs are molecules that are going to be used to synthesize DNA or RNA. The PCR buffer will allow the pH of the solution to stay within the range required by the reaction. Magnesium chloride is used to stabilize some of the free phosphate groups that are going to be found in the solution due to the reaction. And the nucleus-free water, we just ensure that we use nucleus-free water so that it is DNase and RNase free, so that these DNases and RNases are not present to degrade our initial template. Some of the more variable components of RT-PCR are going to be the forward and reverse primers used and the genomic material being used for the reaction. Here we're using a positive sense single-stranded RNA template, and then the forward and reverse primers are going to be designed to target specific sequences. So the specific sequences that we are going to be targeting is the N1 gene, the E gene, and the RNA-dependent RNA polymerase, or RDRP gene in itself. These three genes are highly conserved regions, and if you remember from the previous slide I had of the SARS-CoV-2, the N1 gene is responsible for making nucleocapsid proteins that are bound to the genome, and the E gene is responsible for making the envelope protein. And so these three genes, the N1 gene, the E gene, and the RDRP gene, are all highly conserved among different coronaviruses. Therefore, they have actually been studied pretty well, which makes it fairly easier to detect SARS-CoV-2. So a little bit more about the RT-PCR reaction. So for RT-PCR, you're going to start with the RNA template. So that's from your SARS-CoV-2. That's what you've isolated from the sample. And then this RNA strand has a poly A tail, and in order for transcription to begin, you need to add a primer. So we're actually going to add a poly T primer that will bind to the end of that poly A tail. So once the primer is bound, reverse transcriptase comes in to elongate this strand by adding DNTPs, and then once this occurs, you have created a cDNA molecule, which is a complementary DNA molecule of your RNA. And so once this complementary DNA has been made, specific primers that you have designed to target SARS-CoV-2 can therefore bind to this specific cDNA, and then Taq polymerase will further synthesize the DNA to amplify. So a little bit more about DNTPs. So DNTP, or deoxyribonucleic triphosphates, are these high energy molecules that are used to synthesize DNA or RNA. In this specific example, I am showing how DNA is synthesized. So essentially what happens is that DNA polymerase assists in this synthesis, and DNA polymerase will help cleave off two phosphate groups in order for the nucleotide monomer to be added to the growing DNA strand. However, these two free phosphate groups will be hydrolyzed and into this diphosphate molecule. And in this hydrolysis reaction, energy is actually released, making it a very thermodynamically favorable reaction, which will not allow the reverse reaction to occur. Therefore, it's great. We're creating this DNA synthesis occurs and will not go in the reverse reaction. This diphosphate group in itself is highly negative. Therefore, that's where the magnesium chloride comes in to stabilize and quench this negative charge of the diphosphate molecule. So a little bit more about the primer design. So again, these primers are designed to bind to specific target regions of the template in question. And so these primers will typically be between the length of 18 and 30 nucleotides. This is all going to be dependent on the size of the genome. The larger the genome, the larger you want the primer to be so that it could specifically bind to the region of interest. If it's a smaller genome, you can get away with using a smaller length of primer in order to target that specific region. Again, we are going to be designing primers that are complementary to the target sequence of the N1 gene, the E gene, and the RDRP gene. So another important thing to remember about these primers is that they have to withstand very high temperatures of PCR cycling. So in order to withstand these temperatures, you want to increase that GC content, because again, if you remember, GC nucleotides have 3 hydrogen bonds that bind them together, therefore making them a lot harder to cleave because it would require more energy to cleave 3 bonds that hold the G and the C nucleotide versus the A and the T nucleotide. So these are the primers that were designed. We have to design forward and reverse primers to synthesize the cDNA. So you need the 5' to 3' strand and the 3' to 5' strand. So you will also notice here that we have probes that were designed in order to bind to specific target sequences as well. So these probes are more used for real-time PCR so that we could detect SARS-CoV-2 in real time as PCR is ongoing. So the probe itself has a reporter and a quencher, and if SARS-CoV-2 is present in the sample, that means this molecule will be amplified in the sample. So when this reporter binds to the specific region, as synthesis is occurring, this reporter will be cleaved off and it will release a fluorescent signal. And in order to detect the fluorescent signal, you need to use a specific machine, as seen in this image here. So right here, you will see a thermocycler. So this thermocycler has an optical module that will detect the fluorescence of that reporter. And then here you will see the results of what it would look like after running a RT-PCR reaction in real time. So on the x-axis, you will have the number of cycles that occur as PCR is ongoing. And then on the y-axis, that is going to be essentially the viral load that you will see. So if there is presence of the virus in your sample, you will see this nice S curve occur, because as PCR is ongoing, you will continue to amplify this further and further and further, therefore making the fluorescence the fluorescence signal much, much stronger at an earlier cycle. However, typically when you detect someone with coronavirus, it's if someone has a CT value that it's lower, so like a cycle value that is lower than 20, that's definitely someone who has the virus, because it was present in such a small amount and has amplified so quickly within the reaction. However, people within the 30 range right here, so the 30 range and like 30, like anything below 38 range, this will also be indicative of someone who has a positive result for coronavirus. However, sometimes we have some noise where some people, you might see something further beyond 38. This could be someone who is presenting, who could have coronavirus or who may not, but this is left up for the clinical lab scientists to further determine and to further conduct more tests to determine whether or not someone is positive for the coronavirus. So a little bit of summary and comments. So the accuracy of RT-PCR, um, it will vary and it will decrease as time progresses from the initial infection of the person. So essentially, if a person would want to really know whether or not they have coronavirus, they should be testing quite frequently. And that's the importance of testing frequently. Typically, the accuracy of the test is actually roughly around 90% when the person starts presenting symptoms, which is actually really great. We want to be able to detect it early on so that person can isolate. There are also other tests out there that are used in conjunction with a PCR test, or also independently, but it actually works much better when it's worked in conjunction. And this test is the antibody test. So this antibody test will actually detect whether a person had COVID-19 or not. So this test, however, a lot of research has to go into this test because not much is known about immunology, and essentially there have been ongoing trials, and some people who have tested positive for coronavirus using the PCR test have tested negative for antibodies for coronavirus. And again, this is something that is ongoing. A lot of research is going into it. Because again, this will improve the accuracy of the test. It will allow people to know whether or not they had coronavirus. Another really great test out there that is being developed is next generation sequencing, which it will essentially allow us to test for coronavirus at a much higher volume. Again, NGS, it's much more, it's a much more complicated process, and it will allow a lab to essentially test thousands of people all within one reaction versus the current test, which is the PCR test, which will only allow us to probably maximum test maybe like 300 people. So going into this step of NGS will actually increase the volume of people that we are testing. Thank you so much for listening, and thank you.